Greetings again. So we were cut off a little bit in that last video, but we're looking at the human genome. So trying to understand how is the DNA in human cells arranged. And so a helpful tool for understanding that is this thing called a karyotype. Now, what a karyotype is, it's literally a picture. It's just an arrangement of all the chromosomes in the human body arranged in pairs and organized by size. And literally they would I believe in the early days, they would use a microscope to visualize the chromosomes and then cut them out and arrange them like this. And so you can see they're arranged in pairs, right? The homologous pairs. And the longest chromosome is 1, then 2, then 3, and they get smaller and smaller as you go, all the way to 22. And then X and Y are last because they're this kind of special pair that we'll look at in a moment. So somatic cells are all the cells in your body except sperm and egg, and somatic cells in your body contain 46 chromosomes. So they have all the chromosomes pictured there. They are diploid cells, which means that those 46 chromosomes are actually arranged in pairs. And we call those pairs homologous. And in the previous screencast, I kind of highlighted that if we have this red chromosome and blue, and they're homologous, it means they have the same type of information, but not necessarily the identical information. So these both big G and little g, maybe they code for, eye color or something. Big G might be brown eyes, little g might be blue eyes. And you can see all the way down the list, right? Big G, big R, big S, little t, big T. They have the same type of information, but not exactly identical. And the reason why you have these pairs is because half your chromosomes came from your biological mother, half came from your biological father. Okay? Now this is confusing. So I'm going to go to camera 2, and I want to show you just kind of an analogy for a cell that is diploid, where 2n is equal to 6. So camera 2. So camera 2 is looking at a tabletop here right now, but we're going to add some chromosomes. So this is a cell where 2n is equal to 6. And so for my example, I'm going to use little candy bar wrappers here. And so here we have one particular chromosome. Here's a second chromosome. Here's a third chromosome. Here is a fourth chromosome. And then here is a fifth and sixth chromosome. Now, if you notice, you may notice that there's some similarities in the candy bars here. So you'll see that there are 6 total candy bars, or 6 total chromosomes. 1, 2, 3, 4, 5, 6. But they're also arranged in pairs. You may notice that there are 2 dove bars. These dove bars are similar, but not identical. They both have the dove information, but they're slightly different versions of dove. Same with the Skittles, and same with the Milky Way. And so we would call these homologous because they have the same information. Oh, there's Dr. Hout's calling, that's funny. But are slightly different. Same type of information, but not identical. So back we are at camera 1. So homologous chromosomes. We clear on what homologous means? I hope so. The other thing about these 23 pairs of homologous chromosomes is that there's an additional categorization we have. So one pair of those 23 pairs are called the sex chromosomes. That is the X and Y chromosomes. The other 22 pairs are called autosomes. So everything else is just an autosomal chromosome, and then this last pair is the sex chromosome pair. The sex chromosomes are called the sex chromosomes because they determine sex. If an individual inherits an X and an X, then they will be a biological female. If they inherit an X and a Y, they will be a biological male. So XX is female, XY is male. And then just a little side note, we do use the term sex in biology. Gender is actually more used for social constructs of male and female and may or may not align with biological sex. So if we're talking XX, we would say it is the sex is female, but gender may or may not, depending on how that person identifies. Let's dive in on those sex chromosomes a little bit. Kind of interesting. So the Y chromosome is actually tiny. And it's called Y because you could see this little picture here. It looks a little bit like a letter Y, kind of, sort of. It has very little genetic information. It actually only contains about 100 genes versus about 2,000 on the X chromosome. So relatively little genetic information there. But one really key set of genes there are the ones that trigger the development into a biological male. And so, naturally, that's a biggie. And then the genes that it does have are homologous to those on the X chromosome. So even though they're different sizes, we would still say they are homologous to one another. A little bit more on the X chromosome. So females have this interesting situation. So males are XY, females are XX. And what that means is that females have this sort of double dose of that X chromosome. They have 2 copies, whereas men only have 1. And that's actually problematic because females, we can't have them getting double dose of all of those genes. And so what happens is this process called X-inactivation. And this only occurs in female, and it happens very early in development. So when they're still developing in the womb. And what happens is, in each cell, one X chromosome, and it's just random which one it is, but one X chromosome inactivates in each cell and condenses into something called a bar body. And so we're going to talk a little bit more about the mechanism of how it inactivates. Spoiler alert, methylation is involved. Not that exciting. But you can see, here we have a zygote. And so if this kind of purple one inactivates, it kind of crumbles up, coils in on itself, and becomes inactive. And then all the and then basically those cells are going to have the purple. If the purple one inactivates, then we're going to be left with the blue. So this cell is a descendant of this cell that the blue inactivated. This one is a descendant of here where the purple inactivated. Why is this interesting? Well, it's kind of interesting because this explains the coloration pattern of a calico cat. So it turns out that the gene for the colors of black and orange are actually located on the X chromosomes. And so if you have a heterozygous cat with one orange and one black chromosome here, what happens is early in development, when that cat is just a ball of cells, one particular X chromosome in each cell inactivates. And so here you can see it's the orange one, here you can see it's the orange one. And so all the descendants of these cells are going to have black pigmentation. Whereas here it's the orange cell, or the black cell that inactivates, and the black cell that inactivates. So the descendants of these cells are going to have the orange pigmentation. White is a whole other gene involved and gets a little more complicated, but the black and orange kind of patchwork pattern is because of the origin of which of those 2 chromosomes inactivated. So that explains the calico cat. Okay, finally, just again, those are the sex chromosomes, and then the autosomes are all the other chromosomes that are not sex chromosomes. I don't have much to say about those. So all of that was about the somatic cells. I want to remind you of the gametes as well. And again, that study guide outline will kind of help you make sense of the order I'm doing things here. So the gametes, again, are just the sperm and egg. They contain 23 chromosomes, meaning they're haploid. So they only have one member of each homologous pair. And so the next question then is, how do we produce these haploid gametes from diploid cells? And that is what we're going to talk about in our next screencast on meiosis.